Hello there. Welcome to the last part of Chapter 12 on neurophysiology. This is part 3, and this entire lecture is all about the action potential. All right, it's really quite a large subject, but we are just going to try to keep it to the basics. All right. When I was studying for my neuroscience degree, I took a year of neuroelectrophysiology. So that means for 2 whole semesters, I got to learn all about the action potential. But I'm just going to present it to you here in 1 week. All right. But that just goes to show how complicated this action potential can really be. It really goes beyond the scope of what we're doing, but you will leave here with, the basic knowledge of what an action potential is and in general what's going to be going on with it. And it really is something that every anatomy student needs to learn about. Students have asked me before, like, why do I have to learn about this? Well, it does pertain to the clinical world. In terms of drugs, for example, there are lots of drugs out there that are targeted at manipulating the action potential in some kind of way. Also, next semester in 139, when you study cardiophysiology, for example, you'll get into the action potential of the heart. And definitely a lot of drugs are designed to manipulate the action potential in heart muscle. So it does have a lot of relevance. All right, well, let's get into it. So we have to first start off with some definitions here. Okay, so neurons, like all cells, have what's called a resting membrane potential. Now, I did bring this up a little bit when we talked about muscle physiology and how the neuron signals to the muscle cell to start contraction. So I did bring this up a little bit before, and I mentioned that, for example, there is a resting state in the muscle cell, and that is represented by a number measured in millivolts. And I don't know if you remember, but for muscle, that number was -70. All right, so now that number can vary based on the cell. All right, so the resting membrane potential, we're going to talk a lot about this, and we're going to talk about how it is maintained. So that will be a high yield area of study for you for this PowerPoint and this lecture, is how do we maintain the resting state? How do we keep our cells at the resting membrane potential? So this is something that you find in muscles and neurons, okay, or skeletal cells. But you should know that neurons are really excitable. They're very responsible to the movement of ions across its membrane, all right, even more so than a muscle. All right, so yeah, neurons can very rapidly change their resting membrane potential compared to muscles and compared to any other cell. Now, opposite charges are attracted to each other, right? And what are we talking about? Ions. We're talking about charges that are attached to ions. And you already know, we talked about it way back in chapter 2, that opposite charges are attracted to each other. Now, the thing is, energy is required to keep these ions from going towards each other. Okay, so let's say, for example, you have a lot of negative ions inside of the cell versus a lot of positive charges outside of the cell. Well, those charges, being that they're opposite, are naturally going to try to come towards each other. Okay, so in excitable cells, there is a level of energy that's going to be required to keep these ions from moving towards each other. Okay, and so these opposite charges are going to be separated by the plasma membrane. Now, once we do have, now eventually these charges are going to start moving towards each other. Okay, so eventually that energy is going to be liberated. Okay, and so this is really where the term potential comes from when we say resting membrane potential. Okay, potential, what's really meant by this is it means that we have a separation of charge. Okay, we have opposite charges separated by a membrane. All right, that means that this system has a potential energy. All right, it has the potential to release energy when these charges start moving towards each other, or when the charges start moving across the membrane. Okay, so potential really just refers to the fact that we have opposite charges on either side of the membrane. So definitions, okay, before we really dive into the action potential, we need to definitely go through some terms. The first term here is voltage. Now, I just mentioned how basically we have this situation of a potential energy that's generated by separated charges across a membrane. Well, this potential is measured by voltage. Okay, so voltage measures potential energy. Okay, and so of course we're going to measure the potential, or the difference in charge that exists on either side of a membrane by measuring both points, right? So we'll measure the inside of the cell versus the outside of the cell, and again we're going to see that measured as a voltage. And this is called a potential difference, because there's a difference in charge across the membrane, okay, resulting in potential energy. So the greater the difference in charge in the two areas, so the greater the difference in charge on the inside versus the outside, okay, the greater that difference is, that means we have a higher voltage. Now, when we have resting membrane potential, the term polarized is used. Cells are considered polarized when they are at rest. You should definitely know this number for sure. There are a few different values that I'm going to ask that you know, and this is the first one. So, like muscle cell, the resting membrane potential of a neuron is negative 70 millivolts. Now, cells, okay, and this really pertains to not just neurons and muscle cells, okay, but all cells, all right, that at rest, okay, during rest, we have more negative charges on the inside of the cell compared to the outside. All right, well, yeah, that's why we have a negative 70 millivolts, because we're really seeing, we're looking at how this measures the difference in charge. Okay, so it's going to, if it's a negative value here, then that means we have more negative particles, more negative ions on the inside, and that is the normal resting state of the cell. Oh gosh, another blurry slide, but I think we can get through it. Okay, so some more on definitions here, all right? There's a second term here that you should know. It's called current. Current. Current simply is referring to the flow of ions. It's the movement of ions across the membrane. Okay, that's all current is. Now, current, you can really break it down and analyze it, and there are a bunch of really complicated equations that we could get into that involve looking at how voltage, current, and resistance, which is our third term, are going to change. All right, there's a lot of mathematics involved in that. Okay, but for our purposes, we're just going to know that current is referring to the flow of ions, okay, or the flow of electrical charge across the membrane. All right, so when we're dealing with cells, you know, a lot of times we're talking about sodium current, okay, because sodium is a big deal ion that we're going to be dealing with here, as well as potassium. You can say that there's a potassium current or a sodium current. That just simply means that that particular ion is moving across the membrane. Of course, we're going to have channels, right? We're going to have ion channels. We're going to have sodium channels, potassium channels, etc., that are gated, all right? We're going to have voltage-gated sodium channels, for instance. We'll have voltage-gated potassium channels, all right? And of course, these gates on these channels will open or close based on various stimuli, but in our case, we're really going to be dealing with voltage, so it will be voltage that causes these gates on these channels to open or to close, to open or close. All right. So, current depends on channel activity. All right. Current, okay, whether or not an ion is going to flow into or out of a cell obviously depends on whether or not its channel is open or closed, right? So the condition or the configuration of a channel at any given moment is going to dictate. Okay, and again, we're really dealing with how voltage changes will cause these channels to open or close. We will deal with a different channel, which is not gated, and we've actually mentioned it before. It is it's going to be a potassium channel that's always open. It's not gated. We'll bring that up again. All right, our third term here. Okay, we dealt with voltage, current, and now our third one here is resistance. And you all know what resistance is, all right? It's something that hinders something else, right? So in this case, we're talking about a hindrance to current. Anything that hinders the flow of ions is considered a resistor. All right, so some variables that are going to dictate resistance is whether or not there is an insulator present. Now, if any of you have studied electricity or know anything about electricity, like how it runs through cables and wires, this really is related to that, right? So an insulator, you should know that an insulator is something that will provide a high resistance, okay? There will be a high resistance, meaning that there will be a resistance to the charge flow across something, in our case, a plasma membrane. So an insulator is going to provide high resistance, meaning it will hinder current. Okay, it will hinder the movement of ions. So I'm just going to go ahead and mention here that myelin, which I talked about a little bit, myelin, which is that thick fat-based substance that is found around axons. Myelin is considered an insulator, okay, because, well, for a couple of reasons, all right. Mainly, well, it's lipid, right? So lipid insulates. So wherever there is myelin in an axon, there will be a hindrance to the flow of ions across that area wherever myelin exists. All right. Remember the nodes of Ranvier. Remember that the nodes of Ranvier were these little sections in between patches of myelin that did not have myelin, right? They were devoid of myelin. The nodes of Ranvier are bare. There are parts of the axon that are not myelinated. So the nodes of Ranvier have a very high density of sodium channels. So the current that we see flowing in and out of an axon is going to be through the nodes of Ranvier, okay? Not through myelinated parts of the axon. Okay, and this is important for the conductance overall of the action potential, because wherever it's myelinated, we're keeping these ions from getting out of the neuron, out of the axon, so that we can continue to have that action potential propagate all the way down to the end of that axon. Okay, so main idea here is, what is resistance? And understand that myelin is something that provides resistance because it is an insulator. Okay, now what is the opposite of an insulator? A conductor. A conductor is something that has a low resistance, all right? Something that would not hinder the charge flow, all right? So again, degree of thickness of myelin is going to contribute to the resistance of neurons. So these definitions bring us to an equation that y'all need to know. There will for sure be a question on it. I'm not going to be putting numbers here. I just am concerned that you understand the relationship, and that's the same like with a lot of your math classes, things you learn in math. I think a lot of the times the instructor doesn't necessarily want you to they want you to understand how the variables, how the numbers relate to each other. What's the relationship between the values. And that's the same thing here when we talk about this equation that has to do with the action potential, okay? This definition, or I'm sorry, this equation is based on something known as Ohm's Law. And maybe you've learned about this before. Definitely in physics is something that you deal with. So Ohm's Law gives this relationship between voltage, current, and resistance. And like I said, you should definitely know this. So current, which you can abbreviate means current, is equal to voltage divided by resistance. Okay, definitely want to know that equation. And you really want to know the relationship here between these variables in this equation. Okay, so current, which is the flow of ions, is directly proportional to voltage. Okay, so this should make sense because the greater the voltage, remember that voltage measures potential difference. Voltage measures how different the charges are on either side of a membrane. Okay, so voltage measures that. So if you have a greater difference, okay, in the charges on either side of a membrane, you have a greater difference in charge, that is going to be a major drive for the flow of ions, okay? The greater the difference in charge across the membrane, the greater drive start moving, okay? So voltage is directly proportional to current. So if you have high voltage, this means you're going to have high current, all right? If you have a low difference in charge across each side of the membrane, then there is no major drive or motivation for those ions to start moving. So a lower voltage would mean a lower current. Current, however, is inversely proportional to resistance. This should also make sense, all right? And I mentioned it on the last slide. The greater resistance you have, okay, we're talking about what is resistance? It's going to hinder ion flow. So if you have a high resistance, you're going to have a smaller current. Okay, they're inversely proportional. So you should definitely know how these variables relate to each other and this equation. All right, so a little more here about ion channels. And I think you already know this stuff. This is really review. Okay, so ion channels existing in the membrane, they are proteins, of course, and they're very selective for what they transport, all right? A potassium ion channel is only going to transport potassium, for example. And then you have two main classes. You have leakage or non-gated channels and gated. Remember that the leakage channel, we're really dealing with the potassium leak channel, and this is really important in understanding the action potential, very important. The gated channels are going to include voltage gated. Now, we do have three kinds of gated channels. You have chemically gated, voltage gated, and mechanically gated. Chemically gated, we've dealt with before. Remember acetylcholine with muscle physiology? Acetylcholine is released from the neuron, and that chemical, ACH, binds to an ACH receptor, okay? And then that receptor serves as a sodium channel. Okay, so that would be an example of a chemically gated sodium channel because it opens or closes in response to a chemical binding to it. Voltage gated, all right, voltage gated is really the big deal about the action potential, okay? That's what we're going to be dealing with primarily in this lecture. Mechanically gated, we're not really dealing with. All right, and here's just an explanation of what was already said. Okay, and again, here's just a picture of this, all right? Chemically gated ion channel. Again, look over here to the left and you'll see this is actually a sodium channel, but it has a little receptor site on it. And we're going to have a neurotransmitter, all right? When we say chemical gated channel, we're going to be talking about either a neurotransmitter or a hormone for the most part. Okay, so neurotransmitter comes over here and binds to the receptor site on this sodium channel. That causes the sodium channel to open. Well, this is actually showing a sodium/potassium channel because you do have channels like this. This is not the sodium-potassium pump. This is not the sodium-potassium pump, so please don't get confused. All right, you do have channels that will, antiporters. Remember, you have channels that will transport more than one type of substance in or out of that channel. But again, this is not a sodium-potassium pump. Totally different. Kind of similar, but different. All right, voltage-gated channel over here on the right. All right, so this is voltage-gated. So right now we're showing that this sodium channel, here's the little gate, and it's closed. But what we see over here, look at the charges. See, on this side there are positive charges. Over here we have, and now it's more negative, okay, on the outside and more positive on the inside. That change in the voltage is going to cause this voltage-gated sodium channel to open in this case, and that's going to allow sodium to come in. All right, so how do we measure voltage? Well, we use something called a voltmeter. All right, this is going to measure the potential across the membrane. Now, let's talk about resting membrane potential. Remember I said resting membrane potential will be an important topic here. Okay, now why does our resting membrane potential exist? It exists because of this potential charge difference. Okay, it exists because there is a difference there. All right, so there's a difference in electrolyte distribution between the extracellular fluid and the intracellular fluid. Resting membrane potential is negative 70 millivolts, remember. So the inside of the cell, the cytoplasm, is more negative compared to the outside. We can also say in this situation that the cell is polarized. And again, how is this potential generated? How is this difference at rest generated? Well, it's generated by differences in the ion concentration in the ICF versus ECF at rest. Also, differences in plasma membrane permeability. Now, differences in plasma membrane permeability, that is dictated by whether or not channels are open or closed. Make sense? Okay, whether or not something's going to go through the membrane, which is permeability, okay, whether or not a membrane is permeable to something is totally dictated in neurons by whether or not channels are open or closed. Here is just a little picture of a voltmeter. All right, you just stick this electrode in there, okay, into the axon microelectrode, and then we get a value there. Oh gosh, I hate when these slides get blurry like this. Okay, so, and this is kind of an important slide actually. I'm sure that you will need to know this information. So remember, we're talking about rest, okay, and one of the things that's going to help maintain resting membrane potential is the fact that we will have differences in ionic concentration at rest. So you should definitely know for sure that at rest, the extracellular fluid has a higher concentration of sodium compared to the inside of the cell. Okay, at rest, there is a higher concentration of sodium outside the cell. Right? So think about that. Remember when we talked about muscle and we talked about how sodium rushes into the muscle cell and that excites it. All right, well, that's why it excites it, because it's not normal to have a whole bunch of sodium inside the cell. So when you do have a whole bunch of sodium coming into the cell, that is what excites the cell. That's what excites it, okay? But at rest, all right, we're going to have more sodium outside the cell. And the counterpart is that the intracellular fluid has a higher concentration of potassium compared to the outside of the cell. Okay, so at rest, we have a higher level of sodium outside. At rest, we have a higher level of potassium inside. Okay, now, you know, again, we have this situation where positive and negative charges will want to attract to each other. Okay, so this is sort of balanced out on the outside of the cell when it comes to sodium with chloride. Chloride is the negative ion outside the cell, which kind of helps keep sodium out there. Okay, remember, positive and negative attract. Okay, so at rest, we'll have sodium outside the cell, and the negatively charged chloride, which is outside the cell, is going to help sort of keep that sodium out there, right? Sodium goes together with chloride. Now, as far as helping to keep this potassium higher on the inside, that means we're going to have some negative charges on the inside that are helping to keep that potassium inside at rest. And really, this is because of all the negatively charged substances that you find inside of cells. Okay, we have a lot of large proteins that don't leave the cell. They stay inside the cell, and they're negatively charged. Something else that's negatively charged that you're going to find a lot of inside of the cell is going to be phosphates. Remember ATP? All right. The phosphates that are attached to ATP are negatively charged, and the ATP stays inside the cell. Okay, so that's going to help keep that potassium inside at rest. Okay, so keep in mind, this is the situation at rest. Okay, now another thing. Potassium, when you think about resting, when you think about, let's say, for example, we just had an action potential. Well, we have to end that action potential somehow. And that is going to be caused partly by efflux of potassium. Efflux, meaning potassium will leave the cell. Okay, because what are we having happen during an action potential? During an action potential, we're having an excitation of the neuron, and that is caused by sodium influx. So to end or stop the action potential, we need to bring the cell back to a negative resting state. So to do that, one thing that the cell is going to do to bring the neuron back to a negative state on the inside is it's going to get rid of positive charges that are located inside of the cell. Okay, so we're going to use potassium and its positive charges, and we're going to have the potassium leave the cell to return the neuron back to a resting state. Okay. Now, something else, really important point here, is that the plasma membrane of a neuron is more permeable to potassium than any other ion. And you should know why that's the case. Remember, what does it mean when we say that a plasma membrane is permeable? It means that that membrane is open to receiving things or letting things out. So what dictates permeability? What dictates permeability, and I already said it, is whether or not ion channels are open or closed. Okay, so the deal is that in the neuron, we have a much higher number of potassium leak channels than we do sodium leak channels. So this is why the neuronal plasma membrane is more permeable to potassium compared to sodium or any other ion. All right, because what's going on with a leak channel, potassium leak channel? A leak channel is always open. It's always open, always. So there's always a leakage of potassium. And now the leak channel works one way. We're going to have potassium leave. Okay, the leak channel transports potassium out of the cell. Okay, so important point there. All right, that's going to help you understand how we maintain this resting membrane potential. Part of it is due to the fact that we have more potassium leak channels than any other type of leak channel. So we're always continually going to be having potassium leave the cell just sort of at a slow but steady rate. And that is part of what contributes to maintaining that negative 70, that negative value, because we're having positive charge steadily leave, okay, due to these potassium leak channels. So this is a really important slide. definitely should have an understanding of this for quite a few questions that you'd have about the action potential. This slide just says what I already said. All right. As far as we do have large negatively charged proteins that because they're large, they're not going to be leaving or really entering the cell. So those negative charges will stay inside of the cell. And again, that partly contributes, okay? Just the fact that we have these large negatively charged proteins inside of the cell, that also helps contribute to maintaining that negative 70 millivolts. And then again, because we have more potassium leak channels, all right, that's another reason. And remember, we're doing one-way transport with these potassium leak channels. Potassium is leaving the cell. We're having potassium efflux. What else helps maintain the resting membrane potential? Well, I already mentioned, more potassium is leaving the cell compared to sodium coming in because of potassium leak channels. This helps establish and maintain the resting membrane potential. Now, what else helps maintain the cell at rest at negative 70 is this all-important sodium-potassium pump. Remember what we're doing here with that sodium-potassium pump. Okay, so let me make something clear on this. When we are returning the cell to rest, potassium plays a huge role in two ways. Okay, when we're trying to bring the cell back to rest after an action potential, potassium, first of all, is going to be leaving through voltage-gated potassium channels. So you're having potassium efflux through voltage-gated potassium channels, and you're having potassium efflux through potassium leak channels. Okay, so those two right there, all right, this is important. Those two right there, voltage-gated potassium channels and potassium leak channels. Those two types of potassium channels are allowing potassium efflux. So through both of those types of channels, we're having potassium leave the cell. We're having positive charge leave the cell in order to bring the cell back to a negative value inside. Now, the sodium-potassium pump, I want you to understand that the sodium-potassium pump helps maintain the resting membrane potential. It plays only a small role in returning the cell back to rest. It has a greater role in, after it has been returned to rest, maintaining that resting state. Okay, so there's a subtle difference there. All right, so sodium-potassium pump is really important in maintaining, stabilizing that resting state. Once that resting state has been reached, due to the activity of the voltage-gated potassium channel and the potassium leak channels. Okay, I hope you all see that subtle difference there. All right, so reminder on this sodium-potassium pump, what is it doing? It's pumping 3 sodium out of the cell and bringing 2 potassium in. This helps explain why we have a higher concentration of sodium outside the cell at rest because we're taking more sodium out. This explains why we have higher potassium inside the cell compared to outside because we're bringing potassium inside through this sodium-potassium pump. So maybe you can see then why the sodium-potassium pump is not going to play a significant role in bringing the cell back to rest. It plays a significant role in maintaining this resting state once it has been reestablished. So this is showing you, okay, look out here. This is the extracellular fluid. And yes, at rest, we have a higher concentration of sodium on the outside compared to the inside. On the inside, we have a higher, at rest, concentration of potassium compared to sodium. That's really all it's showing you. Okay, another thing. Now, remember, we don't have as many sodium leak channels as we do potassium leak channels. All right, we have many, many more. There's a greater density of these potassium leak channels compared to sodium leak channels. Okay, so this is why we're not going to have a lot of sodium flow in a sodium leak channel compared to a potassium leak channel. Okay, but keep in mind, just that, our potassium leak channel is open all the time. So although we're having potassium efflux through that, remember what else we have here that's not shown: the sodium-potassium pump. So just as soon as we take this potassium out through the potassium leak channel, we're going to be bringing some of that potassium right back in through the sodium-potassium pump. Okay, so again, that sodium-potassium pump is going to play a big role in keeping our potassium levels high inside at rest. Well, here's your sodium-potassium pump, just as a reminder. All right, we're going to be taking sodium out, 3 sodium out per every 2 potassium in. This also explains why, okay, the inside of the cell is more negative at rest, because we're removing 3 positive charges on that sodium compared to 2 positive charges we're bringing in on that potassium. Okay, so that's another explanation for why we're able to maintain that negative 70. And this one is just a summary, again, of these different channels and what's happening. Now, this up top is showing you potassium leak channels. All right, so we're going to have potassium leaving through our leak channels. This is at rest. Remember, we're still talking about the resting membrane potential. The second one down here shows the fact that it's trying to illustrate that, yes, we do have more potassium leak channels in the membrane compared to sodium leak channels. All right, it's showing here's 2 for every 1 sodium channel maybe. All right, so again, at the end of the day, we're having more potassium leave the cell through potassium leak channels compared to the amount of sodium that's coming in through sodium leak channels. So I know I mentioned that potassium leak channels are taking potassium out. You should just note in comparison that sodium leak channels bring sodium in. Okay, so but again, we're having more potassium leave. We're having more positive charge leave than we're having come in. Again, that's what partly keeps our negative 70. And then, of course, we have a very important contribution to maintaining rest with our sodium-potassium pump. All right. So when does our potential change? Remember what potential is. It means we have a difference in charge across the membrane. So it changes when concentrations of these ions, particularly sodium and potassium, if concentrations of these ions around the membrane start to increase or decrease. Okay. Once that happens, it's going to trigger the activity of channels in the membrane. Okay, and so that's when the permeability of the membrane will start to change. Okay, so concentration of ions or potential is going to dictate whether or not channels are going to open or close, and that's going to dictate current. It's going to dictate whether or not we're going to be passing ions across that membrane. All right, so 2 types of potentials that we have. We have action potentials, all right, and we also have something else, which I mentioned before, but didn't want to get into the details of it too much before, but we will talk about it now or sometime in this lecture. Graded potentials. So graded potentials, all right, we have these type as well. So we have 2 types of potentials. Now, mostly what we're dealing with are action potentials. Action potentials are capable of traveling a long distance. A long distance. So this means these will take place in long axons. Okay, that's one fact. What about graded potentials? Graded potentials are going to travel over short distances. Short distances. Okay, so here's what happens. All right, you have a neuron. All right, and let's say it's a postsynaptic neuron. It's going to be picking up signals from a presynaptic neuron. So the dendrites on the postsynaptic neuron receive signals from other neurons. When that happens, there are these electrical signals, okay, or potentials that emanate from the dendrites and go to the soma. Remember the anatomy of the neuron. Dendrites are the receiving part. The dendrites will pick up an electrical signal, and that electrical signal will like emanate around the soma. Now, that is considered a graded potential. The electrical signal coming in from dendrites, they are not action potentials, okay? The electrical signals coming in through dendrites are classified as graded potentials because they only travel a short distance. They travel from the electrical signal travels from the dendrites and goes to the soma. Now, here's what happens is you get a whole bunch of graded potentials. They're small electrical signals traveling over this short distance from dendrite to soma. You get a whole bunch of these and you add them up. Graded potentials get summated. They get summed up. Okay? They're going to be added together. And if there are enough graded potentials to create a strong enough electrical signal, then this will trigger an action potential. All right? You remember the part of the neuron called the axon hillock. The axon hillock was that area where the soma and the axon intersect. It's at the axon hillock that if we have enough graded potentials that added up there in the soma, and right there at that axon hillock is where we will see an action potential begin. Okay, so the action potential begins at the axon hillock if we have a strong enough collective signaling from these graded potentials there. So basically, all of your action potentials are preceded by a number of graded potentials. Okay, so you have to have enough of these occurring at the dendrite and soma in order to cause an action potential to develop at the axon hillock, and then we can carry that signal all the way down to the end of the axon. It will not stop. It will go the whole distance. All right. Let me just see something really quick here. Yeah, okay. So the action potential, how are we going to change the resting membrane potential to cause an action potential to happen? Well, we're going to have something known as depolarization, which I already mentioned that with muscle physiology, what that means. It's referring to excitation. It's an excitatory event on the cell. Okay, and what happens in depolarization? It is marked by sodium influx. Be sure to know your terms, influx versus efflux, because that's the way it's going to be termed on the exams. So sodium influx is associated with depolarization. This is what causes excitation of the cell, right? Because at rest, we're negative inside the cell. So if we have a whole bunch of positive charges rushing into the cell at a high rate, then that is going to excite the cell. We then see that the inside of the cell becomes less negative. Or you could say the inside of the cell becomes more positive. Now we will see that with depolarization, we will see that negative 70 go all the way up to positive 30. Positive 30 millivolts is the peak of the action potential. And you should know that number too. Okay, so we're going to go from negative 70 up to positive 30 millivolts inside of that cell. All right, we're going to go up to positive 30 due to the influx, massive influx of all these positive charges associated with sodium. That's depolarization. Now, what is repolarization? I think that I mentioned that before as well. Repolarization is when the cell is attempting to come back to rest. Okay, it is reached once we reach our peak millivolts, which I already mentioned is positive 30. Now, it really depends on the cell. Maybe one neuron is, maybe its peak is positive 35. But an average standard number is going to be positive 30. So we're going to stick with that. Okay, so once that cell reaches positive 30 millivolts on the inside, that triggers the cell to start to do this process of relaxation. Repolarization is when we're trying to repolarize a cell. We're trying to bring it back to a polarized state. We're trying to bring it back to rest. This is largely characterized by potassium efflux. Okay, what we're going to see is potassium efflux through voltage-gated potassium channels. All right, now I know I mentioned potassium leak channels. Okay, but potassium leak channels, yes, they do play a role in repolarization, but the real initial stages of repolarization, the first thing that's going to happen, or one of the first things, is really the opening of voltage-gated potassium channels where we see efflux of potassium to get rid of that positive charge inside the cell to bring the inside of that cell back to a negative 70. Now, there's another little phenomenon that's going to occur during the action potential, and it's called hyperpolarization. This is considered an inhibitory event, and it will be important for you to understand what is causing hyperpolarization. So hyperpolarization is caused by, well, what is it first? Hyperpolarization is when the inside of the membrane becomes just a little bit more negative than resting membrane potential. So in other words, it can go all the way down. As we're trying to repolarize, we can go all the way down to negative 90, right? For a very, very brief time, we're actually going to go even more negative than our normal negative 70, and we're going to go down to about negative 90 millivolts for an extremely brief period of time. All right, and the reason that is the case is because these voltage-gated potassium channels are slow to close. Okay, what we're going to see is that potassium, the channels associated with potassium are slow versus these voltage-gated sodium channels, which are really fast to open or close. Okay, so but as we're repolarizing, these voltage-gated potassium channels, they're slow to close. They close very slowly. So that means we're going to dip down just a little bit lower than negative 70 for a very brief period of time until those voltage-gated potassium channels end up closing. Once those voltage-gated potassium channels close, then we no longer see potassium leaving the cell from those channels, and then this will help bring us back up from a negative 90 to the normal, regular negative 70. Okay, so on this illustration, what we're seeing is we're seeing time here on the bottom, and up here we're looking at membrane potential measured in millivolts. Negative 70, that's your resting state, resting state. Okay, so this line will just be straight, just level here. Now, during depolarization, we're having sodium influx. You will see an elevation in this line. Depolarization is seen as an elevation in this line because we're going more positive here. We're approaching what's known as threshold. You should know that threshold must be reached in order for the action potential to completely reach this positive level up here. All right, negative 50, negative 55 is threshold. Okay, we have to have enough depolarization to take us to negative 50 or negative 55 in order for that action potential to proceed all the way to its peak. Now up here it's showing you positive 50. All right, you may have neurons that where their peak is positive 50. all right? But positive 30 is a good standard number, again, for peak. All right, so that's kind of, you're seeing an elevation in the line here with depolarization. Over here on the right, if you're looking at hyperpolarization, well, remember, negative 70 is resting. And for a very brief period of time, while those voltage-gated potassium channels are still open and you're still having potassium efflux, remember, those potassium channels are slow to close. So we're having the inside of that cell go just a little bit more negative than what we would see at rest. And so you would see then a depression in this line here. Okay, we're going to see a dip down with hyperpolarization. And remember, that value is usually about negative 90. All right, I'm trying to determine if I want to continue here. Give me a second. Yeah, let me just do a few more slides and then I will stop this video. And we'll have a part 2. All right. Yeah, so I think I already explained this, okay? Graded potentials. Remember that graded potentials they only go a short distance. They only act over a local area, okay? And they don't travel a long distance because they're short-lived. All right? Remember, the stronger the stimulus, okay, or the stronger the stronger the stimulus in any event, the more voltage is going to change, and the farther we're going to go with current. In the situation of graded potentials, we don't have this situation where we have one strong graded potential. You have to have a lot. It's all about the number of graded potentials. That's what it's about. Okay. If you have a lot of graded potentials, then we can get this situation where we're able to trigger an action potential, and we will get the flow of current consistently down through the rest of the axon. All right. I've really already said this. Something else, you can consider the fact that graded potentials, the signal will dissipate. The signal decays. And that makes this very different in definition from an action potential, because an action potential does not dissipate. It does not decay. An action potential starts and it goes all the way. It doesn't stop. Okay, so it doesn't die out. It doesn't fade. The signal doesn't fade away or dissipate. But in graded potentials, this does happen. The electrical signal will fade away or decay. Okay. And then, like I mentioned, you have to have enough graded potentials summate, all right, or collect at the soma in order to cause this action potential to start at the axon hillock. And, this is just showing you the direction. Here's a reminder. You remember your neuron anatomy. You have your dendrites out here, the receiving portion, right? And it's showing, like, here's a dendrite close up down here. The dendrite has all of these sodium channels and potassium channels in it. Yes. Dendrites are not myelinated, remember. Okay, so they're going to have a lot of sodium channels present. And so, yeah, we're going to have depolarization of our dendrites. Okay, and these will result in graded potentials. Now look at the short distance between the dendrites and the soma, short distance. Okay, but we're having ion flow. We're having sodium rush into these dendrites and rush in, but that's only going to last so long in a graded potential. If it does reach the soma, okay, if we're still having sodium current into the soma and we have enough depolarization happening here from all this sodium, we have enough of this, we're going to summate all that and add it up. And then if it's enough here at the trigger zone, which is also known as the axon hillock, then we will get the development of an action potential. And that action potential will go the entire length of the axon. All right, it will not stop. It will go to the end here and cause all sorts of activity here at the synapses. This is just showing you like a hypothetical axon here, all right, and how, let's say we have a stimulus, we have some electrical signal coming to a dendrite, for example, all right, so here it is, all right, it's just going to excite this small area, all right, and then what you'll see is that the depolarization will spread a little bit. Okay, so it'll spread. Okay, that means we're having the flow of sodium inside of this dendrite. Okay, this could also be an axon. All right, because certainly some axons do very short axons will just do graded potentials. All right, but in any event, you're going to see that the signal spreads. Okay, so we're having positive charges sort of spread along the inside of this membrane, causing the inside of the axon or dendrite to be more positive, okay, where we're getting that sodium influx. Okay, now, but what you're going to see is that it dies out. Okay, it's not going to last a long distance if it's a graded potential. It will eventually die out. Okay, and that's how this is shown on a graph here with the membrane potential and versus distance. Okay, distance is not going to be long. It's just going to sort of decay out if it is a graded potential. You know, that's might be a good place to stop, because the next part I'm really going to go into detail with the action potential, but I think you have a really good basis right now for us to get into that on the next video. All right, so I'll see you for part 2 of this next time. Have a great day.